Gas exchange primarily controls the respiration rate. The goal of respiration control is mainly focused on how well the exchange of gases is achieved by the lungs. Although the rate of oxygen and carbon dioxide production by the body cells vary widely with changing metabolic demand, respiration has to be controlled so as to maintain appropriate levels of oxygen and carbon dioxide. This mechanism depends on the regulation of ventilation through the interaction of neurological and chemical control mechanism. This respiration mechanism is controlled by various factors like neurological, chemical and other factors. All these factors are interconnected with each other directly or indirectly. Let us see all the factors in detail. Neurological factor. Breathing is controlled by a poorly defined group of neurons in the brain stem called the respiratory center. The components of this center are widely scattered throughout the pons and medulla oblongata. As seen in the figure, this widely dispersed group of neurons, collectively called the respiratory center, can be divided into three areas on the basis of their functions: the medullary rhythmicity area in the medulla oblongata, the pneumotaxic area and the apneustic area in the pons. Let us see medullary rhythmicity area. The function of the medullary rhythmicity area is to control the basic rhythm of respiration. It is a part of the central nervous system situated in the medulla oblongata. The medullary rhythmicity area is also known as the respiratory switch center. There are inspiratory and expiratory areas within the medullary rhythmicity area. Inspiratory area. Nerve impulses generated in the inspiratory area establish the basic rhythm of breathing and is also responsible for the contraction of the diaphragm. The inspiratory area sends signals to the diaphragm via the phrenic nerves. During quiet breathing, inhalation lasts for about 2 seconds and exhalation lasts for about 3 seconds. Let us see expiratory area. Neurons of this area usually remain inactive during quiet respiration. However, during forceful expiration, for example during heavy exercise, it is activated by nerve impulses from the inspiratory area. Impulses from the expiratory center cause contraction of the internal intercostals and abdominal muscles, resulting in a forced expiration. Let us see pneumotaxic area. The pneumotaxic center is also known as the pontine respiratory group, abbreviated as PRG. It is located at the center in the upper part of the pons that provides inhibitory impulses on inspiration and thereby prevents over distension of the lungs and helps to maintain alternately recurrent inspiration and expiration. The major effect of these nerve impulses is to help turn off the inspiratory area before the lungs become too full of air. In other words, the impulses shorten the duration of inhalation. Breathing rate is more rapid when the pneumotaxic area is more active. It can be considered as antagonist to the apneustic center. Now, let us understand apneustic area. Apneustic area in the lower pons also coordinates the transition between inhalation and exhalation during breathing. This area sends a stimulatory impulse to the inspiratory area that activates it and prolonged inhalation occurs. The result is a long, deep inhalation. It controls the intensity of breathing. The apneustic center is inhibited by pulmonary stretch receptors. When the pneumotaxic area is active, it overrides signals from the apneustic area. Disorders involving abnormal stimulation of apneustic center can produce a gasping type of ventilation with maximum inspirations. Regulation of the respiratory center. The basic rhythm of respiration is also influenced from other brain regions, receptors in the peripheral nervous system and other factors. Chemoreceptors. In our body, we have sensory neurons that are responsive to chemical stimuli, which are termed as chemoreceptors or chemosensors. These receptors are divided into two main groups based on their anatomical location, and each has its own pattern of sensitivity to regulate respiration. First is central chemoreceptors and the other one is peripheral chemoreceptors. These chemoreceptors are influenced by monitoring levels of carbon dioxide, H+ ions and oxygen. Central and peripheral chemoreceptors mainly function to regulate respiratory function. This is an important mechanism for maintaining arterial blood partial pressure of oxygen, partial pressure of carbon dioxide, that is PCO2, and pH within appropriate physiological ranges. Central chemoreceptors are located in or close to the respiratory center in the medulla oblongata in the central nervous system. They respond mainly by changes in H+ ions or PCO2, or both are less affected by changes in arterial pH or PO2. As seen in equation, carbon dioxide, that is CO2, reacts with water, that is H2O, to produce H+ ions. This in turn results into a drop in pH that directly stimulates the central chemoreceptors. An acidosis of the arterial blood has little effect on central chemoreceptors. Additional input for respiration processes comes from peripheral chemoreceptors that are located in the carotid bodies, which are oval nodules in the wall of the left and right common carotid arteries, and the aortic bodies located in the wall of the arc of the aorta. These chemoreceptors are sensitive to changes in H+ ions, PO2 and PCO2 in the blood. Peripheral chemoreceptors are less important than central chemoreceptors in the responses to an increase in PCO2. The central chemoreceptors are stimulated and respond vigorously to the resulting increase in H+ level. Chemoreceptors monitoring CO2 levels are actually sensitive to the H+ level liberated by the dissociation of carbonic acid. The signal are transmitted to the aortic bodies by the part of the vagus, that is 10th nerves, while the right and left glossopharyngeal, that is 9th nerves, are sensitive to the carotid bodies. Nerve impulses from the hypothalamus also stimulate the respiratory center, allowing emotional stimuli to alter respiration, as for example in laughing and crying. Proprioceptor stimulation of respiration. Proprioceptors that are sensory and organs in muscles, tendons and ligaments that are sensitive to movement of joints and muscles. As we start exercising, our respiration rate and depth increase even before changes in PO2, PCO2 or H plus level. The main stimulus for these quick changes in respiratory effort is input from proprioceptors. The proprioceptors stimulate the inspiratory area of the medulla oblongata. Herring-Brewer reflex. All the air conducting pathway, as in the cardiovascular system, from the bronchi to terminal bronchioles has smooth muscle. The Herring-Brewer reflex, based on smooth muscle of the bronchi and bronchioles, affects depth of respiration. The smooth muscle of the conducting pathways has stretch sensitive receptors called baroreceptors or stretch receptors. When these receptors become stretched during overinflation of the lungs, nerve impulses are sent along the vagus nerves to the inspiratory and apneustic centers. Due to that, the inspiratory area is inhibited directly and the apneustic area is inhibited from activating the inspiratory area. As a result, exhalation begins. As air leaves the lungs during exhalation, the lungs collapse and the stretch receptors are no longer stimulated. Thus, the inspiratory and apneustic areas are no longer inhibited and a new inhalation begins. This reflex, called as the inflation reflex or the Herring-Brewer reflex, is mainly a protective mechanism for preventing excessive inflation of the lungs. Let us see chemical control. Chemical factors modifying respiratory center activity. The respiratory center is also regulated by a number of chemical factors which control the depth and rate of respiration. Arterial blood gases and pH. Blood gases play a vital role in the regulation of ventilation as they are monitored by the respiratory chemoreceptors. Arterial PCO2 is most important, with pH playing a secondary role, allowing respiratory compensation for metabolic acid base disturbances. Elevated arterial PCO2. Respiration is stimulated by any elevation of arterial PCO2, leading to a compensatory reduction in alveolar PCO2 as excess CO2 is blown off. This is the primary mechanism responsible for the regulation of breathing and ventilation. As we see in the CO2 dissociation curve, regulation of CO2 levels is important since any rise in PCO2 increases the CO2 content of the blood and promotes acidosis. Lower level of CO2 in blood cause acapnea, resulting in the cessation of breathing. Acidity. Fall of blood pH can stimulate the respiratory center. Acidosis occurs by increased CO2 that lowers the pH, that ultimately enhancing the rate of breathing. The condition is reversed at the time of lower CO2 levels. Depressed arterial PO2. Levels of arterial PO2 is also important in regulation of ventilation. Low levels of arterial PO2 can also stimulate respiration. PO2 must drop very low to about 40 mmHg in order to stimulate respiration as seen at high altitudes. However, if CO2 in the alveolar gas is increased, O2 can stimulate ventilation at a PO2 higher than in normal condition. If the O2 does not reach the blood, asphyxia occurs. Reduction in the supply of O2 to the tissue causes hypoxia. If the tissues completely lack O2, it leads to anoxia. Other controls of respiration. There are other factors that also help to regulate respiration like vasomotor center. Vasomotor center is primarily responsible for cardiovascular management, but also stimulate breathing, for example, air hunger in surgically shocked patients. Temperature. An increase in body temperature increases respiratory rate, for example, fever. Pain. Pain causes the heart rate, breathing rate and blood pressure to rise. Usually, when a person is in pain, the limbic system as well as the sympathetic center in the hypothalamus gets influenced. This causes the respiration rate to increase, but a sudden severe pain brings about brief apnea and absence of breathing. Emotion. Emotions involve physiological changes within the entire body. Breathing is primarily controlled for metabolic and homeostatic purposes. However, breathing can also change in response to changes in emotions such as sadness and happiness. Irritation of airways. Certain substances such as smoke and pollen, medical conditions and medicines can irritate within the nasal passages or airways, can induce coughing and sneezing. Blood pressure. A sudden decrease in blood pressure increases the ventilation rate, while the condition is reversed in high blood pressure. Blood pH. Sensors called the aortic and carotid bodies detect changes in blood pH. The lungs and kidneys collaboratively control blood pH. An abnormally low blood pH increases the respiratory rate. Rapid breathing releases increased amounts of carbon dioxide, a major determinant of blood pH. Various drugs. Certain drugs such as narcotic pain relievers and sedatives and the illicit drug heroin can depress the activity of the respiratory rhythmicity center in the brain stem. High levels of alcohol have the same effect. So, the nervous system, with the help of other dynamics, normally adjusts the rate of ventilation almost exactly to the demands of the body, so that the oxygen pressure and carbon dioxide pressure in the blood are hardly altered even during heavy exercise and most other types of respiratory stress.
In this part, we have studied the control of respiration, which is a complex and multifactorial process. In this part, we have studied the control of respiration, which is a complex and multifactorial process. We have listed the major medullary and pontine areas that control respiration and briefly mentioned their role in ventilatory control. We have also learned role of various factors like pH, partial pressure of carbon dioxide, oxygen partial pressure, temperature, pain, blood pressure and etc. In sum, we can say that the breathing can be modified by input from cortex, the limbic system and the pattern of breathing can be influenced by emotion and by various diseases. The main aim of respiratory control is to maintain appropriate homeostasis of the blood gases and facilitate proper oxygen delivery to the body.